So we'll continue with a small presentation by me for terrestrial sampling and Stella for aquatic sampling. I am Elisa, and okay, this is going to be just quite an introductory talk of how to sample in general. I'm sure there are a lot of people here more experienced than me to Malaise traps and setting Malaise traps. And that's why, okay, I will keep it general at first, what we need to think when we are sampling. And afterwards, please feel free to make any suggestions, etc. I'm just going to provide a backbone of what is considered best practice, etc. And then, of course, we can have either discussion, okay, it's sampling, or afterwards when we prepare the guidelines of all this procedure, maybe you can, of course, suggest anything you believe that it is worth saying. So, so as I said, I'm going to start talking general for sample collection. Due to the diversity of taxa and the variation of habitats and ecosystems species live in, there are for sure a lot of different things to take into account when you are preparing to go sampling. And finally, after being involved in several sampling campaigns, let's say, or sampling efforts, then actually you, let's say, earn a skill, and which is actually even, it can get even further developed, a skill to sample a specific taxa or specific species. So I'm just going to, as I said, just present the basic steps you should consider when you're before you go for sampling and during your sampling. First of all, you have to design your sampling, what you want to collect, of course, where to find what you want to collect, the species, and how to collect it, the gear that you're going to use. And in some cases, when you should collect, depending if you have different developmental stages or species that live periodically. Afterwards, you go to the actual collection of the specimens, how many you're going to need. So in order to see how many you're going to take out from the habitat. And of course, if you need to repeat the sampling. Another thing that you should also take into account is that all this info that should accompany the specimen, the metadata that is needed in order to, let's say, fully not identify, but characterize your sample. The minimum required data on bold is, of course, an ID, where you got the sample. If it's a museum, okay, the museum voucher, but if it's a phrase, then you don't need that. The institution where the sample is stored, and of course, the taxonomy of the species and where did you find it, in which country. Afterwards, we have the documentation of the specimen, which means that a photograph will work even with your mobile phone, where the specimen size and colors are easy to be shown, and why that? For future, let's say, reference for those that are going to identify again the specimen, who want to identify the specimen. The specimen should be preserved. Ethanol is usually used in animal tissues, of course. And then the specimen is identified. It means it's morphologically, it follows the morphologically taxonomy it follows. And if you want to make DNA extraction, you just take a piece of the tissue of the specimen in order to continue with DNA extraction. Since this whole workshop is under the umbrella of BGE, which is a huge project, who among others, but mainly focuses on arthropods and especially insects, which is something expected since there is a lot of DNA barcoding information missing from this taxa. And you can actually see that from the gap list as it was published in GBOL. So you can see that the categorization of the specimens of the species were done as arthropods and non-arthropods, and insects and non-insects. So we're going to focus on this group of organisms, of species. There are two approaches to collect arthropods, not only arthropods, but we're going to talk about that. There is the active collecting, which requires a more physical effort using nets, aspirators, and the passive collecting, which involves the setup of traps. You can see here different kinds of traps, pan traps, pitfall traps, malaise traps, and of course, when you select the gear that you're going to work with, you somehow, well, not somehow, by selecting your gear, you're actually selecting, you make a bias, let's say, you select actually the group of taxa that you're going to also to collect. We will continue with malaise traps, explaining what malaise traps are. They are tent-like traps. They have short-hand walls and one big wall, one central wall. These are all black. And the roof is white, a white net. And here on the top of the roof, there is a collection tube, which is usually filled with preserving fluid. The insects, they go towards the malaise trap, and they hit the middle wall. So those that fly upwards, they fly toward the peak of the trap and get into the jar where is the preserving liquid. So the malaise traps are easy to, not easy, well, they're very, a lot of people, entomologists who work with them, because you may set them for a long period of times, and they should be, of course, checked weekly or week after week, depending on the area maybe and the season. And there are some taxa that are, of course, there are a lot of flying insects, a lot of species trapped in the malaise trap, but Diptera and Hymenoptera are the two taxa that are mostly trapped in this kind of traps, malaise traps. Where they can be set, forests, open grass areas, and generally in areas where you expect to find insects. They are more they work better in sunny areas than the shaded areas. And there is also a study that, not only one, but that it seems that it should be orientated north to west. Before you set a malaise trap, you have to check also the vegetation around the trap in order not to have any obstacles against the insect flight to the trap. And of course, check for high winds. These are just a few things that you have to check. Under the umbrella of BGE in Arthropod University of Thessaloniki, and is involved in this high mountain arthropod system study, where malaise traps have been set in 5 different altitudes of here in Vermio in Greece. They are set from June to October for 5 months. Samples are collected weekly. This is done by the School of Agriculture, Dr. Koveos, Dr. Koulousis, and Anastasia Kokari and Yorgos Floros, who actually is with us. And he's more expert than me to talk about malaise traps. And those of you that are going to continue in Crete in the conference of entomologists, you'll also meet Anastasia Kokari there. So what we expect from this experimental setup, a bottle is going to be sent for DNA metabarcoding in order to see how communities of flying arthropods change across different altitudinal gradients in high mountain systems. Not only in Greece, it's in Vermio, but this is an action, this is an experimental setup followed by many other partners of the BGE. And the other bottle of the other Malaise Trap is going to be checked for species that may be used for filling in the gap of the gap list of DNA barcoding. And of course, some samples are going to be sent to Sage for duck biodiversity sampling. And that was me for Malaise Traps. And for, let's say, general steps for sampling.
Hello, and from me, I'm George Floros. I'm postdoc researcher in agriculture faculty. I'm working here with Elisa and Spiros and the other guys for all this big program. We use, as Elisa tells you in an excellent way, Malaise traps to capture some insects about our process in the program. Mostly Diptera and Hymenoptera insects that can fly, and Malaise traps do flight interruption, and the insect follow the route of the trap, you will see how, and goes inside a bottle we have, and we keep them there with some alcohol or other chemical we can use. We use alcohol, but mostly because, okay, we change Malaise traps weekly, every week, but usually we want to use them in their best way. We have to leave them for 2 weeks and maybe more, so that we will collect more insects. In that situation, we don't use alcohol. We use a mix of water with some soap, so that the insects will go in the water.
Did you try glycol also? We are using this 50-50 glycol solution.
We tried not in Malaise trap. We tried in pitfall traps.
Yes.
We used also pitfall traps. By digging the ground, we put a bottle there with this solution exactly, and we had not a lot of insects captured there, not a lot of insects. Some beetles and some very, very small arthropods, right? Not insects. Hylopoda, myriapoda, and stuff like that. Yeah, exactly. So, Malaise trap. I have to say that Malaise trap is not just like this shape, the tent trap or this color. In Malaise traps belongs a lot of trap kinds with different shapes and different colors. But this is the one that the most of entomologists use because it's the most effective one. Unfortunately, this work requires two persons to do it. Is a tent. We have to open the tent. We have to take the skeleton. We were lucky enough because these Malaise traps have very good skeleton, because there are some Malaise traps with very bad quality of skeleton and easy breaks. And we start from the front side. The front side of the trap is the one who has the bottle. Inside here, we collect the insect. Κράτα λίγο. Περίμενε. Κράτα αυτό. Α, μπράβο, αυτό. Αυτό ήθελα. Ναι, κράτα μπορώ. And after that, it's easy. Because the only thing you have to do is to click the fabric in the skeleton. Ναι. Oop. This is the difficult part. Κάτσε Σπύρο μπροστά. Όχι, κάτσε να το βάλουμε πρώτα και μετά. And this is our trap. It's a big tent who needs definitely soil to keep it stable.
So I know how things those are actually size of the tent matters much, or what they're studying, because I never.
The size
The size matters. That's good.
Yeah. Rain of it.
Yeah,
Rained
of course.
yesterday and
Course.
that kind of stuff, yes. But I guess that
Actually, the color matters in no way. As well. You get
Yeah. But you mean with color or
I'm not. It's not my area. It took me an expert who's a instructor. He's not been involved in that.
So this is the trap. We are very careful when we place the trap in the field to understand which is the direction of the wind. We want all this side to be the wind to come directly here. So all the flying insects will come here, there they will stop, and because the insects, the flying insects, have the chance to go up and fly up, they will start again flying and walking up, and they follow this route until they reach to the only gap the trap has, which is this bottle. And inside this bottle, they get trapped. So every week we go, we change, we have this. This is the half of it, about the half of it, with liquid alcohol or the solution of liquorine, whatever. And every week we just go and we change this bottle. If you want, your trap to be in the field for a long period, you have to be very careful when you will place this in the soil. This is the most important. You have to place it in such distance Thank you for your assistance. In such distance that you will need no hand to keep it, but only this. Okay, so you are very careful when you put this in the soil to put it until there in the top, and to be very careful to make it like, like this. Okay, so no wind, no animal, no person will be able to take it with you, except it's a beer. Yeah. Here even there were periods with strong winds, especially the periods now of autumn, because we have north winds in the area, are okay. But we were lucky enough also because all around the spots we placed it, there were kind of trees. So it will be some of protection there. And of course you can enhance the efficacy of a Malaise trap. Just not only put the tent in the field, but you have, if it's possible, an extra machine, a light, even with battery, blue light or white light, not red light, because insects do not see the red light. And you can put the small light somewhere here, and in the night also to attract the insects, so you have attraction from the light, and when the sunlight come out, they will walk and fly to the gap here and be in the bottle. That's all. That's a Malaise trap.
Excellent. Thank you, George. Thank you very much. And now we go back inside with Stella to talk about aquatics. Huh? Okay. So.
Hi everyone, I'm Stella Minoudi. I'm a postdoc researcher here in AUTh. I work in the group of Alexandros Triantafylidis. Today I will talk to you about aquatic sampling with vampire sampler. Yeah, this is the vampire sampler. I have started working on eDNA projects relatively recently. So I think that there are people here that are more experienced than me. So please, everyone, feel free to share your knowledge, your experience to this presentation. I think that most of us are familiar with the term of eDNA, environmental DNA. We are talking about the DNA that can be extracted from environmental samples, for example, soil, water, or sediment, without prior isolation of the organisms themselves. eDNA is a mixture of genomic DNA from many different organisms, and it is possibly fragmented. Possible sources of eDNA include sloughed skin cells, urine, saliva, or other body parts. As regards the potential eDNA applications, maybe we talk about the detection of single species, developing single species approaches, maybe by using qPCR, or we are talking for biodiversity surveys, so as to find out the community composition by using meta-barcoding. This is what is used for BGE, and for this is necessary to produce the barcodes, which is what we are doing here in this workshop. Ways of water sampling. Depending on what we have to do, what we want to do, there are different approaches. If we need to take samples from deeper depths, maybe we can use the Niskin water sampler, which is the first one that we can see there. Or we can use simple ways, like a sterile collection bag or bottle, or a peristaltic pump that will pump the water to take the samples. Then we have to filter the water that we have collected. Again, there are different ways for doing this. We can use a vacuum pump or a peristaltic pump, or a simple approach like a disposable syringe. Today I will present you the vampire sampler, which is this one. Okay, this is a peristaltic pump. We have the drive unit, the pump unit, and we put the hose in this hole. It provides many advantages when we use this because it is portable. We can handle very easily this and carry it to the field. There are two different batteries which provide us the time to take all the samples we want. We can take samples from even 5 meters depth. We can limit the contamination risk because we can, the only disposable thing that we use is the hose that we have to sterilize it before we use. And finally, we can simultaneously filter our sample. So, when we want to use it, we just open this one. We put the hose, and in the one end, we adapt the filter. Okay, there are different types of filter. And we put the other side into the water. Okay. And we start, press the on-off button. Okay. We have to check that the water goes to the right direction. If it is not the right, there is a button that changes the flow. So I will do it.
Okay, okay. Well, normally it's in the water.
Yeah. Yeah. That is very simple. When you finish to filter the volume of water that is needed, or maybe until the filter is clogged and no other volume is can be filtered, just release the on-off button, and you have finished with the filtering and taking your sample from the field. And then the next step is just to take all of the water volume from the filter, and then you preserve as you want. If you want to keep it a long time and do the next steps after a long time, you can add a preservation buffer inside it, or otherwise you can still keep it in the freezer and let it thaw until so as to proceed with the DNA extraction. Okay. If anyone wants to try it, it is here. Yeah.
I don't want to try it. Now, I was just wondering, so then it means that, do you clean when you go on the filter, it means that you have a lot of different tubes?
Yeah. It's important to sterilize all the hoses you use, and it's important to have a different hose for each sampling site, so as to avoid any contamination between the sampling sites. With bleach solution, and rinse it with distilled water.
So the DNA is inside
The filter, yeah.
and then you break it to just, or how do you
No. I proceed all here, inside the filter.
The DNA extraction, how do you access the filter?
All the solutions come inside the filter, and then you take the DNA. Yeah. Yeah.
How many liters you can filter by this?
It depends. We have succeeded in filtering with this kind of filters around 1.5 liters, but it depends on the environment and how it is. The minimum volume that I succeeded to filter with this is 400 milliliters.
Do you use also protective mask or just the glove when you are doing this?
Just gloves. Yeah.
And what about controls, negative controls?
Yeah, also negative controls are needed. When we prepare a bottle of distilled water in the lab, and then we filter it in the field.
So
Again with a clean hose.
you are filtering water straight from the pond, for example, or you are doing some pre-filtering from
Most of the times we do it directly from the field. Yeah.